Okay, today we're talking about isotopes, and I want to give you a brief introduction to isotopes, you as the TAs. This is not something you'll do with the students necessarily, but you're going to bring this knowledge in throughout the lab. So I want to make sure you're really clear about what an isotope is and what the isotopes are that we're talking about in this lab. Okay, and so an isotope is an atom that differs in the number of neutrons. Alright, so in this lab we're looking at carbon-12. So if you remember from chemistry, and the students have learned this in chemistry, that carbon has an atomic number of 6, right, and it's got a weight of 12. And so this is the normal and the most abundant carbon isotope on the planet. And so if we were to draw that atom, its atomic number is 6, which means that it has 6 protons in its nucleus. 1, 2, 3, 4, 5, 6. Okay, and then because it weighs 12, then the other weight is neutrons. And so you can tell from this that it also has 6 neutrons. 1, 2, 3, 4, 5, 6. Okay, and then it's got 6 electrons that surround the atom. And I'll just draw those. We don't really deal with the electrons in this lab, but you're going to have 2 in the first shell, 4 in the outer. Alright, so there's carbon-12. And the difference that we're looking at carbon-12, carbon-13, carbon-14 we're not looking at in the lab, but those are the 3 most common isotopes. So carbon-13 will just have 1 extra neutron in its nucleus, and so its weight would be 13 instead of 12. Okay? And so that's what we're looking at, and it's a stable isotope, whereas carbon-14 that has 2 extra in there is actually radioactive, it decays, it's unstable and it decays, and so we're just looking at carbon-12 and carbon-13. And so it turns out that the majority of the atmosphere of the carbon on the planet is carbon-12. That's the most common isotope. But there's a little bit of this carbon-13 and even less of the carbon-14. And so what scientists did is they looked at a marine fossil, and they took the amount in this fossil, they took the amount of carbon-13, and they divided it by the amount of carbon-12, and they came up with a ratio number, and it's what? 0.0112372. Did I get that right? 2372. All right, so that's going to be their control, their standard amount of carbon-13 to carbon-14. So there's very little. You can see it's a very small ratio. But let's say if you look at a plant material or animal tissue, and it has even less carbon-13 than this marine fossil did, then it's going to be a number that's smaller than this, and it's going to be a negative number. If it's got more than this, it would be a positive number. Now we are only dealing with negative numbers because it turns out that photosynthetic organisms, when they bring it in, they much prefer the carbon-12, and so any plant or animal material is going to have way less carbon-13 than in this ratio. So we're only dealing with negative numbers. But the smaller the amount, the even more negative it is. So if you had a pile of carbon-12, right? So let's say this is the marine fossil. What do we call it? PDB. Okay, this is the marine fossil, and this is how much of that carbon-13 it has. Okay, and that makes this ratio. Then if you were to look at plants, if we're looking at a normal C3 photosynthetic plant, so these are plants like most of the plants you see growing outside, the trees, the bushes, most of our crops, so wheat and barley and potatoes and all of those kinds of carrots, they're all a C3 plant, and so they've got about this much, okay, a very small amount of carbon-13. And so compared to this, they're very, very negative. And so they're on the order of 22, so negative 22 to negative 30 to negative 30, and this is per mil. So when we write this is percent. When we write it with 2 little zeros under there, it's per mil. So that means it's millions of times less than this. It's negative 22 to negative 30 per mil of carbon-13 to carbon-12. If you were to look at a C4 plant, so if we scooped up their carbon, and this is a C4 plant, so these are things like grass, sugarcane, corn. There's just some plants, and remember, if you remember from photosynthesis, C4 plants are ones that live in usually drier climates, and so they don't want to open their stomata, And so instead, what they do is they hold their breath when it's hot and dry outside, and they just bring in the carbon and they store it in 4 carbon molecules like oxaloacetate, and then they bring that into the Calvin cycle later. So it's separating Rubisco from the carbon gathering process so that we don't lose water when we don't need to, and we don't photorespirate. So I don't know if this is all ringing a bell. If you need a quick review of that, you should go back and look at some of my videos. Anyway, so C4 plants, because they have a different enzyme that grabs carbon out of the air and stores it in these 4 carbon sugars, and because of some of the metabolic changes, they will keep a little more of the carbon-13 in their tissues. So they have a little bit more of the carbon-13, and so if we were to look at it compared to, this is the marine fossil, right? And let me see if I can zoom in a little bit so you can see that better. Turn it. Okay, so if this is the marine fossil right here, and that's how much carbon-13 there is, and this is a C3 plant, then a C4 plant is a little bit more than the C3 plant. Okay, so it's closer to the marine fossil, and so as a consequence, it's a little bit less negative than this is. And so you have your carbon, your C4 plants having between -10 and -14 per mil. -10 to -14 per mil. Okay, so they have more of the carbon-13. So they have, this is carbon-13, right? So they have more of it than the traditional C3 plant, and still way less. In fact, this should probably be even bigger, right? So way less than what a marine fossil, what our standard is, but they have more than these guys. And so this is where it throws students and TAs and me half the time when I'm talking about it, is that I want to look at these numbers that are bigger, 22 and 30, and say, oh, that's more. But remember, we're in the negative. So this is -22 and -30 is a much smaller number than -10 and -14. Okay, so the larger the number, or the less negative, the more likely it is to be a C4 plant rather than a C3 plant. Okay, so that's your carbon-13, and so yeah, this is what you need to keep. Oh, gosh, this is what you need to keep in mind right here. Right, is that if it's a more negative number, it's a C3 plant. If it's a less negative number, it's a C4 plant. Okay. Now I'm going to do nitrogen. Let me zoom back out and reset this. Okay, whoops. All right, so we'll draw nitrogen up there in the corner. Okay, so then let's talk about nitrogen. The 2 isotopes we're dealing with in nitrogen are nitrogen 14 and nitrogen 15. So normal nitrogen is 14, all right? So in this case, the nitrogen 15, so nitrogen 14, has 7 protons and 7 neutrons, all right? Nitrogen 15 has 7 protons and 8 neutrons. And so you have a slightly different weight here as well. Now with nitrogen, it's a little bit different. We have a standard, but the standard is 0. So the vast majority of nitrogen in our atmosphere is nitrogen 14, is the normal nitrogen. And so you have very, very little nitrogen 15. And so we actually compare it to the atmosphere. And the atmosphere is basically, if you took nitrogen 15 and you put it over nitrogen 14, you would get 0, meaning there's 0 nitrogen 15. It's not 0, but it's really close to 0. Let's see. All right, because the nitrogen 15 is usually in materials that are not in the atmosphere. So the atmosphere is at 0. And so then plants, when they take that in, they are also at 0 because they're bringing whatever nitrogen they're taking in. Let's see, so atmospheric nitrogen is 100% nitrogen 14, so we have 0. And this is the standard. And plants rely on nitrogen fixation almost entirely from atmospheric nitrogen. So they're not doing it, right? So don't give students the idea that plants are taking nitrogen in through the air. That they have nitrogen fixing bacteria and fungi in the soil that fix that nitrogen, but they're fixing it from the air. And so the nitrogen that's being incorporated into plants is almost entirely nitrogen 14. So if you are a plant, any of these plants, your nitrogen levels are going to be 0 per mil. Okay? But when we eat them, so when we eat a plant, our kidneys and our filtration system preferentially filters the normal nitrogen, which means the nitrogen 15 is sort of like this oh, what do I do with this? It's sort of leftover. And so the nitrogen 15 instead gets incorporated into the proteins in our bodies. It gets incorporated into our muscles and our connective tissue and all of the things that we build. It gets more of this nitrogen 15 incorporated into it than the nitrogen 14 because our kidneys have a harder time excreting the nitrogen 15. So it bioaccumulates. And so what that means is that when we consume a plant that's 0, and it's effectively 0, it has a little bit of the nitrogen 15, when we consume that plant, the nitrogen 15 accumulates. So if you are a consumer, let's say we're doing primary consumption, so we're eating plants, a primary consumer is going to collect a little bit of that nitrogen 15. So over time, if you were to look at my tissue, if I was a vegetarian and I only ate plants, if you were to look at my tissue, you would see that I've accumulated a little bit of nitrogen 15, and it's on the order of about 3 per mil to 6 per mil, okay? So 3 to 6 per mil, right? But then, if you are a cow, or if you are a human who's eating a cow, there we go. So this is the cow, right? The cow's eating the grass, and so the cow tissue is going to be that. But as a human, if I eat that cow, I'm starting at this 3 to 6, and then I'm preferentially excreting the 14, and I'm accumulating even more 15. And so me, as a secondary consumer, I'm going to have on the order of 6 to 10 per mil in my system because I've been eating a protein source. And then let's say you are a fish eater. If you eat a lot of fish, then what you're doing is you're eating the organism, the other organism that ate the plant. So fish often eat, insects. And so a fish is actually one step higher on the food chain than a cow is. And so if you eat a lot of fish, let me just erase that, and you're a tertiary consumer, then you're going to be creeping up to, 9 to 14 on the nitrogen scale. Okay? So this is just bioaccumulative over time, and it's per mil, all right? And so if you look at the trends that I've got there on your instructor guide for you, if you have low carbon-13, meaning more negative, right, less carbon-13 in there, you're more likely to be a high vegetable or native vegetation intake. If you are less negative or a higher carbon-13, you're more likely dining on sugar and corn products. And then for the nitrogen, the higher the nitrogen proportion, the higher up on the food chain you are, the more likely you are to be eating meat products, fish products. Whereas if you're really low in your nitrogen, you've got a really low protein consumption, or you're a vegetarian or something like that. All right, so those are the trends with the isotopes. I want to make sure that you guys understand this before you go into the lab and you've got it solid down in your mind. Okay, so that you don't make the mistakes that I commonly make when I'm doing this lab and I get confused with the students. We don't want that happening. So even if you have to write a little cheat sheet or write it on your palm, what the numbers are, and just always have that with you so you don't get turned around by the lab. Okay, but in the lab, you're not going to give that introduction to the students. They are going to jump right into the data and do Table 1, Part 1 first before you go through any kind of explanation. All right, so I'm going to do that next.